presentation in my email. You know where the slide to send? Okay, this morning we are going to discuss some of the basics of qPCR. How many of you have hands-on experience on qPCR? Please raise your. How many of you? Very few hands are raised. Okay. This side, nobody? So you're going to raise your hand. More strong. Okay. So the purpose of this lecture is only to tell you some of the basics of how the PCR technology really works. The basis of right? You have to change them up. You cannot imagine any transcriptome work or gene expression analytics without qPCR as a tool.
You can directly connect, man. This you directly connect. No? ये तो बजेगा सर इसी पे कनेक्ट किया है सर प्लीज कनेक्ट करने से ये तो it was originally it was connected, no? कनेक्ट हो जाएगा।
सीधा करेक्ट कर लो, व्हाट इज द प्रॉब्लम?
ये से करना पड़ेगा और ओवर से ये कनेक्ट करना इसमें ये कनेक्ट करना होगा फिर कितना मजा रहता है काय करके This is also because then only it will come in. इससे होगा इससे
It's connected. Yeah, it's coming. Okay. Monitor is not connected. Maybe you can. Take some more time. We know this. It's online is going on. Make it fast. First line. But I don't know the presentation there. No? I don't know the presentation. Presentation is here only. what you witnessed is a beautiful example for that. Okay, a couple of lessons from this. Not only your presentation should be ready, but also the gadgets which is going to display your content also should be ready well ahead of time. Okay. Yeah, let us get into the presentation. Probably we may take another 15 minutes. It may go into 11:15. This is very basic content about real-time PCR. All of you must be knowing how PCR as a technology has transformed biological science. I always consider this PCR invention or discovery of this technology or invention of this particular technology as a major milestone that happened sometime in 1980s. So you can conveniently divide the whole biological research into two major categories. One is the pre-PCR research and post-PCR research. So the science what we have been practicing in biology before 1985, you can say that it is a pre-PCR era science. And we have witnessed how much progress was made. And post-PCR era also made a lot of progress in a fast pace that finally led to another era in biology, what is called as the genomic era. So I prefer to call this the whole biological research arena or area into three major categories. One is pre-PCR era, post-PCR era, and genomic era. So if you ask me, the genomic era has tremendously benefited from one technology, that is PCR. So let's see how this technology came into existence and how it transformed into different technologies over the period of the last 20, 30 years. The presentation is all about qPCR and basics. Basics in the sense how this technology is actually working and what are all the principles. If you look back the science from 1970s, although we claim that 1985 was a time when the PCR machine started coming into science, but the foundations and the principles and the proof of concepts, people were working right from 1970s onwards. So this is some of the points what I thought of telling. In 1970 was a time when the first report of replication of single-stranded DNA from template using synthetic primers and DNA polymerase. In 1970s itself, people have given a proof of concept for in vitro synthesis of DNA. So that was perhaps the first major milestone. Somebody has proven that DNA can be multiplied. I would call it amplification, the modern terminology, right in 1970. And if you again go through the literature, 1976 was another major timeline when famous enzyme DNA Taq polymerase was discovered. That was in 1976. In 1983, the people started putting all things together, and a concept for rapid amplification and the synthesis of DNA is experimentally confirmed in the laboratory. And in 1985, whatever information and whatever proof was there in the literature is bundled into a machine, and that machine, what we call PCR machines, are kernel cyclists. That happened in 1985. So who is the person who contributed? I think all of you must be knowing, and Gary Mullis. So 83 was the time when he came out with a proof of concept for that. And very soon, the company RBI, they came into picture, and they started making the machines which can give you different temperature regimes for in vitro synthesis of DNA. So that was in 1985. 1989, again, the magazine Science, they called this enzyme as a molecule of the year. So for the simple reason that this enzyme can really transform the biological science by helping or assisting polymerase chain reaction. And 1993, the whole invention, Gary Mullis was awarded Nobel Prize. So that was some of the historical account about PCR. What I'm talking about, a conventional PCR or the traditional PCR, what we have been practicing in 1980s. And if you look very carefully, this nucleic acid amplification and the detection techniques are one of the most valuable tools in biology. And of course, every branch of biology, we use this PCR, right? There is no branch which we cannot use PCR. And the PCR technology can really accelerate the data generation process, whatever you are doing in biology. So be it biotechnology, medicine, forensic science, agriculture, and every branch of biology. I'm not singling out anything in this particular list. And but if you think very carefully, the conventional PCR, what it is capable of doing is, we will come to know what is getting amplified. More of a qualitative information we were bothered in particular time. So for several applications, the qualitative nucleic acid detection is good enough. So the pathogen detection is one of the classical cases, whether the pathogen is there or not, right? What we call the PCR-based detection, where the qualitative information is more than sufficient. But there are other applications that demanded how much of amplification is there. So the quantitative element, when it comes into the minds of the people, then people started thinking and they started developing a technology, that the technology what we are going to discuss today, that is a quantitative real-time PCR. And there are several names for this, and real-time PCR is one of the very famous name. And of course, people acronymed as RT-PCR, and sometimes it get confused with another RT-PCR, what is called as a reverse transcriptase PCR. Then there is a lot of confusion in science again. Finally, the qPCR is one of the generic term for this kind of technologies, right? So qPCR, the Q is normally written as a small letter and PCR is a capital letter. So those who are not too familiar, try to understand this. And real-time PCR can be used both qualitative as well as quantitative analysis. So what the normal PCR could not offer, the real-time PCR has filled the gap. That started bringing the quantitative elements of the whole PCR into a single experiment. So, of course, we will come to know a little more in the future slides. The history of real-time PCR is as much exciting as the PCR history. So let us see some of the historical accounts, right? The proof of concept, once again, for real-time PCR or quantitative PCR, that was made in 1993 by a scientist, Hikuchi. He recognized that the process of PCR could be monitored. So this is a very important thing, because the reaction can be monitored. Don't imagine that you are going to monitor how different molecules in the PCR mix is going to interact among each other. It is not the case. Rather, the endpoint of the PCR can be monitored. What is the endpoint? There is accumulation of the amplified DNA, and this accumulation of amplified DNA is something which we can monitor through a kind of parameter. Those parameters are what is called as raising graphs in the screen. So let's come to that, how it is really done in future slides. What he suggested was, if amplifying DNA can be tagged with some kind of dye, if we can monitor the dye, and the progressive accumulation of the dye in the PCR mix can be very conveniently captured, and it can be visualized. So that was a very simple concept. So you can see here, Hikuchi, he recognized the process of DNA can be monitored by addition of a fluorescent label that binds to the accumulating PCR product. So very simple concept, but a greater application in biology. As the concentration of PCR product increases, what happens is the intensity of the dye is increases. So this is some kind of a positive direct correlation. So as the DNA concentration increases, the dye what was there in the reaction mixture, which is going to bind with the DNA, is also going to be increased. So the measurement of the fluorescence is actually the basis of real-time PCR, right? And in those years, the dye what was used in real-time PCR is none other than ethidium bromide. So we all know that ethidium bromide is, it can intercalate with the DNA, and that is thoroughly exploited in real-time PCR in those years, right? And of course, this particular discovery in 1993, that paved the way for the modern quantitative real-time PCR. What we are going to do today in the practical is entirely depending on this kind of principles only. So the modern quantitative real-time PCR or qPCR, the only difference is we don't use this ethidium bromide any longer. Instead, we use another dye, which is very safe and not having any the so-called toxic parameters. So the dye what we are using, anybody? The dye what we are using in real-time PCR is cyber green. So we are going to do that today. And 1996, different kind of variants have come to monitor the amplifying DNA. The Tachman Pro, we'll be having a very lengthy discussion here today. So the Tachman detection method, which uses instead of ethidium bromide or other forms of dye, this Tachman detection method also can be used in monitoring the amplification. 1996, the same company that gave the machines, the PCR machines, they again came to market with the real-time PCR machines or qPCR machines. So that happened in 1996. So if you look back, the timeline is very quick. In 1985, we started seeing PCR machines in the laboratory. In a matter of another 10 years, we started seeing real-time PCR machines in the laboratories. So this is a kind of technological advancement in biological research. So the RBI, they introduced the first real-time PCR machines. At that time, these machines were called sequence detection system, and the instrument is ABI 7700. So this is the machine, what it looks like, right? Just take it forward. Now let's have a comparison between the conventional PCR and the real-time PCR, right? Can anyone tell in a conventional PCR how many ingredients are there in the basic PCR? Anybody? In that order, I think those who are doing PCR, they must be knowing. So around 6 to 7 basic ingredients are there in any typical PCR reaction, right? You start with buffer, you will be adding some cofactor in the form of magnesium chloride. You'll be having a substrate, that is a DNTPs. To prime the reaction, you put primers, and you put the enzyme, and you put the template DNA, and of course, to make it up, you put some high quality PCR quality water. These are all the basic ingredients, right? That is how the reaction mixture is prepared and subjected to a particular temperature so that the DNA become separated from each other, the two strands. Then the same mixture is subjected to another temperature. Then the primer comes and binds on the complementary region. Then the extension step where you start adding the nucleotides. And that's how the story, 2 to 3 different temperatures is normally used, right? So let's see the difference. In conventional PCR, you are more bothered about what is amplified, right? So that's what we have I think we have a primer designing class in the coming days. The target amplification, the target amplicon size is very, very important, right? Many times that conveys what is getting amplified. Suppose if you are targeting a particular pathogen detection, and you know very well that if, for example, 450 base pair amplicon is coming, a single discrete amplicon in the agarose gel, that indicates that the so-called pathogen is there in the sample. So what does it communicate to you? What does it convey to you? You are more bothered about what is amplified. That is the hallmark of the so-called normal PCR or conventional PCR. But in the case of real-time PCR, in addition to what is amplified, you are also bothered about how much is amplified. So there is a quantitative element is coming into the whole picture, right? So of course, to address that only, we have some kind of modifications in normal PCR, and that gives you an extra information about how much is amplified in the reaction mixture, right? And of course, I'm not going to read it out. I already told there are a variety of applications in biology where you can use real-time PCR as a tool, whether it's a gene expression analysis. I think today's class is all about gene expression analysis, where you will be analyzing um, uh, the microbe-induced defense genes in plant, how this expression pattern varies across a timeline of around 5 days, right? In every 24 hours interval, you can isolate the RNAs, you can convert that into cDNA, and that cDNA can be used as a template here just to know how the expression pattern varies across 5 different timelines for a particular gene in a plant. So we are going to do that from 11:30 onwards today. And of course, we have disease diagnosis and management and variety of other applications. I'm just giving you only a minuscule of what is really possible in biology. Let's get into a little more finer details here. The quantitative polymerase chain reaction, or qPCR, it's a method of polymerase chain reaction that allows to follow in real time the amplification of the target, right? You are able to see whether the amplification is really happening because the dye what is getting integrated with the DNA is going to increase its intensity with time of amplification, right? The real-time PCR is a technique in which I introduce a word here, fluoroprobe, that binds to the specific target region, that is the target of your interest or the gene of your interest, and it produces fluorescence during the PCR. You'll see that in more details in the coming slides. The fluorescence measured in real time is detected in the PCR cycle with a filter fluorometer. Remember, the story is very simple, and we are going to uh, uh, tag, we are going to monitor the whole amplification process through extra attachment in the PCR machine. That is what is called as a fluorometer or spectropluorometer. We'll just come to the next slide, you'll understand. Therefore, the real-time PCR is a PCR machine combined with a camera. I would prefer to call that camera as a spectropluorometer, right? So in other terms, you have a PCR machine and the reaction is monitored by a spectropluorometer, right? And the combination of these two, you can call it as a real-time PCR or qPCR machine, right? So let's see what happens. I think the image is out of focus here. So it's nothing but you have the PCR machine in the bottom, and there are the PCR reactions are going on in the tube. And on top of that, you are going to keep that spectropluorometer. And the spectropluorometer is monitoring what's going on in the tube or what's going on in the reaction mixture. And that is telling you in the form of some images or curve or graph, whatever you call, what's really going on in the tube. This is the basis of this particular real-time PCR. So this is a little more about the inner content of that particular. If you take a section of this real-time PCR machine, you have thermal cycle in the bottom. In the top, you have the spectropluorometer, and that's going to capture what's going on in the reaction mixture, because in the reaction mixture, you put some reporter, right? Remember, you put some reporter. What is a reporter here? That is a fluorescent dye. Right? That fluorescent dye is going to, what do you call, bind with the DNA, and it is going to report to you what's going on in the tube. This is the basis or the fundamental, right? This is, again, let you know, excuse me.
Director is asking you and I to go.
Office. we'll just wind up. We'll come back and we'll continue. the presentation. We'll discuss those points once again. So this is a conventional PCR, what we already told. But in real-time PCR, please pay attention to the one which is in the red circle. You have something extra, much like what you told now, BSA and DMSO. But we have here an ingredient called detector. You may call it as a reporter also. Okay, so scientifically you may call it as a kind of fluorescent dye. Okay, so up to this point, is it clear to all of you, right? But from here on, we will go to the specific detection system, what qPCR machines are doing. There are two broad categories of qPCRs, right? So one, I call it as a non-specific detection using fluorescent dye, right? So I already put it in the title, specific detection and non-specific detection. In non-specific detection, it exploits the power of fluorescent dye, and specific detection that exploits the power of detective probes. And the rest of my presentation is entirely on these two aspects. How does the fluorescent dye-based qPCR works, and how does the probe-based qPCR works, and what are all the fundamental differences between these two, right? This is a conventional PCR with the inbuilt detector is what we call it as a quantitative PCR. Okay. Yeah, there is another name for that. A non-specific detection by fluorescent dye is also called a dye-based quantitative PCR, right? And the other one, the specific detection by detective probe, is also called as a probe-based quantitative real-time PCR. So there are two broad categories of qPCR. One is a fluorescent dye-based quantitative PCR, and another one is a probe-based quantitative PCR. Okay, so what is another name for that? A fluorescent dye-based, a non-specific detection system, and a probe-based, a specific detection system. The title itself reflects the relative differences between these two and the specific characters of those two technologies, right? Yeah. Let's take the first one. This is the dye-based qPCR. Okay, dye is a very broad term, right? It's a very broad term. The dye what is being used in this particular PCR machine is what is called as a fluorescent dye. Okay, so the fluorescent dye, what Hikuchi conceptualized in 1993, is a dye which can intercalate with the DNA. Right? The dye is nothing but ethidium bromide, but when the time passed, people replaced the toxic dyes with the safer dyes. Right? There are several safer dyes are there. The most popular one, I think all of us are using, you are going to use today as well, that is cyber green. Right? So the dyes such as cyber green that intercalate in the double stranded DNA. Please pay attention here. It intercalates in the double stranded DNA, that too in the minor groove of DNA. I think you imagine the DNA helix. There is a minor groove, there is a major groove. Can you imagine that for a moment? So this particular dye, cyber green, is specifically intercalate with the minor groove. Remember, with the minor groove. Okay. So, so what is the advantage of that? So when the PCR is in action, you have a template at a low concentration, usually around 50 nanograms, 10 to 50 nanograms. And that's a starting of your parent DNA, right? In your PCR mix. How much you add? Anybody? 50 nanograms, 100 nanograms, right? So it is anywhere between 10 to 50 nanograms. So that is the basal DNA you are supplying in a PCR mixture as a template, right? So what happens is that is in a double stranded format. So all of you know that need to be converted into a single stranded format. That is a first step. What is the name of the step? Denaturation, right? So what happens in the denaturation? The intertwined two strands will pull apart. They become two single stranded. All right. Now, what is the next step? Annealing. Annealing of what? The primers. Who designed the primer? Either you got it from the literature, that is called copying, or you designed your own, right? So either way, it's fine, right? So copying is, you are respecting somebody else's work. There is nothing wrong in that, right? So, or your own primers, okay? So what happens here? It goes and bind to a specific site in the DNA where you design the primer, correct? So it becomes a localized double-stranded DNA, correct? Then what happens? Then what happens? Extension starts, right? So it is getting extended. So what is happening here? The double-stranded DNA converted into a single-stranded DNA converted into a double-stranded DNA. Now, this double-stranded DNA is localized. Okay, remember this localized double-stranded DNA also will have minor grooves. Okay, now from here you take the story forward. These are all the dyes what is being used in real-time PCR. Don't think that cybergreen is the only one. It's not so. You have a variety of dyes. You have a cybergreen 1, 2, Eva green, LC green, Pebble, YoPro, Cyto family. The list goes. I brought only a few selected dyes what I could gather from the literature. There are many. You can extend the list, right? Let's see what's happening here. What is the property of the dye what we are using? For example, cybergreen. It gets into the minor groove of double-stranded DNA, right? So this is how it looks like. This is a cybergreen chemistry. Is something like this. You have a double-stranded DNA, right? So they become two single-stranded, right? The primer comes here, right? Then it is getting extended. Yeah, in a solution. What is the solution here? The PCR mix, whatever you have in 50 microliters, or sometime 10 microliters, right? So what is the meaning of this 10 microliter, 50 microliter I'm telling here? Anybody? It's the total reaction volume. Can you tell by the by what can be the minimum volume one can set up PCR? 10 microliter, right? So I don't agree to that. 5 microliter also you can do PCR. Okay, you need to be, your hands should be exceptionally brilliant to take a smallest possible volume, right? Anyway, so in a solution, what happens the day 0 or 0 hour, you have the reaction mixture, you have all the basic ingredient plus the detector. What is the detector here? It's a dye. In this case, it is, let us say, cybergreen. Okay, so now it's in a kind of a dynamic motion there in the solution, right? Now you activated that by putting in different temperatures, right? So what's happening here? The cybergreen that binds to the minor groove of the double-stranded DNA helix. Remember, 0 hour, you have double-stranded DNA, you have the cybergreen also in the mixture, but there would be binding of the dye with the double-stranded DNA in 0 hour. Let's take this point, okay? But what happens there is the emission of fluorescence from that, the starting DNA is beyond the detection limit of any machine. Try to understand that point. You got it? I'm going to relate this with a very famous unit what we generate from qPCR machines, the CQ values and CT values. You will relate these two now, right? There would be a basal level fluorescence is there in any real-time PCR at a 0 hour. The only difference is the machine will not be able to capture that. Why? Because the quantum of fluorescence which is coming from the dye which is bound in the minor grooves of that template DNA is extremely small. Correct? Okay. Even that happens in the denaturation step, that will come out. Is it clear? Clear to all of you? Yeah. So now what happens? The cybergreen binds to the minor groove of the double-stranded helix. After the anneal, the primer dye molecule can bind to the double-stranded.
the DNA binding results in a dramatic increase of cybergreen. Why? Because every step in PCR cycle, there is a geometrical increase in the quantum of double-stranded DNA that is getting accumulated in the PCR mixture. It's clear to all of you. As the cycling steps increases, the number of minor grooves which is available for cybergreen to get intercalated is proportionately will increase. Correct? So there would be a point at which the machine can see what is coming out of the machine. That particular point is the one what we call it as a cyclic threshold value. Right? We'll come to that. This is a point, progressive increase in the fluorescence as amplicon accumulates. You agree with me? Right? As the amplicon accumulates, what happens? The number of double-stranded molecule which is getting accumulated in the reaction mixture also increases. Correct? So when that is increases, the dye which is already there in the mixture is going to bind. Right? So the quantum of double-stranded DNA which is getting accumulated in the form of amplification is directly proportional to the so-called the fluorescence emission which is coming from the machine. Okay. Yeah, intensity of fluorescence is measured by spectrophlorometer. Yeah, this is how that machine, I think the previous slide it was not quite clear, and you have that kind of a PCR machine here, and this is a spectrophlorometer which is sitting on the top of that, and it is really monitoring what's really going on in the reaction tubes. Is it clear? The reaction tubes are in the form of 18-12 configuration, 96 well, and a camera is sitting on top of that is just monitoring what's going on in every tube. Okay, so I'm not getting into different capturing methods. We have a CCD based capturing methods, photodiode based capturing methods. I'll keep that apart, but you try to understand the camera is capturing the image of the reaction mixture in a real time. Okay, and what it captures? It captures the fluorescent image or fluorescence which is coming from the tube, right? So on 0 hour, what happens? It's not seeing anything, although there is a basal level fluorescence. Why? Because the machine is incapable of capturing that image because it is too small for a machine to see. Okay, as the fluorescence, as the amplification increases, there is a number of double-stranded molecule increases, and so the minor groove, and that would provide opportunity for the unbound dye in the mixture to go and bind to the minor groove. So with every passing cycle, the unbound dye is going to become a bound dye. Is it clear? So as the step increases and more dye is getting captured by the DNA, correct? Right? And at one point of time, the machine can see what is coming out of the reaction mixture. Okay. We'll come to that point. And advantages are very straightforward, very easy and cheaper, easy to handle, sensitive, effective. Of course, the major disadvantage of this technique is it is non-specific. The dye is not going to bother who is the double-stranded DNA there. It could be from the target what you designed, or it could be a non-target as well. For example, your primer is not so specific, it is amplifying something else which you never wanted. And there is a double-stranded DNA accumulation in the tube, and cybergreen will go and bind. So for that reason, this fluorescent dye-based qPCR is typically non-specific. Okay, come to that. This is clear. The dye-based fluorescent, I think I simplified it, and keep that. Now we are moving on to the second one. Little complicated, but if you follow that, you will be able to understand what it is, right? Probe-based qPCR. Okay, probe-based qPCR. We understood the dye-based qPCR is quite easy, very cheap. The dyes are very safe, right? Everything is fine. But there is an element of the so-called non-specificity. It might bind with a non-specific DNA, it might come in the way of your amplification, and you might be generating a wrong data. So then Of course, those kind of scenario is not tolerated, especially in the case of pathogen detections. You end up in false positives. False positives are very dangerous, right? So it's not allowed in a typical pathogen detection laboratories. So what would be the best way? Go for probe-based. Okay, what is probe-based? What is probe? What is primer? Are they same or are they different? Okay, listen to a few slides, you will understand. The probe-based qPCR, it relies on the sequence-specific detection of the desired PCR product. Unlike cyber-grain-based qPCR methods that detect all double-stranded DNA, the probe-based qPCR utilizes the fluorescent labeled target-specific probe. This is a single large keywords. Try to remember them. Fluorescent labeled target-specific probe. Okay, fluorescent labeled target-specific probe. Remember, this is not primer. This is a probe. At sequence level, both are same. Both are conveying the same meaning. There is some kind of specificity, sequence specificity. So, come to the long story short here. In probe-based quantitative real-time PCR, you need to have two regions in the gene, and they are specific for that gene. One you call it as a probe, another one you call it as a primer. The one you call it as a probe, you are going to label it with a fluorescent dye. So that is a departure from a normal way of doing qPCR. In cyber-grain, you don't do that. In cyber-grain, it's only one primer. In probe-based qPCR, well in advance, you are designing the primer and also a specific region in the gene that you are going to call it as a probe. I don't mind, you can call it as a primer 1 and primer 2, or even probe 1 and probe 2, no problems. But one of them, you are going to, maybe the upstream of them, you are going to label it with fluorescent probes. Okay, now how to do that? You need to have the prior information about the sequences. You understand? Now come to the main point. You are going to label it with a fluorescent dye. Okay, let's see how it works. Yeah, there are many probes are there. TAC1 is the most popular one. You have a molecular beacons, you have scorpion primers, you have hybridization probes. Please extend your knowledge by extra readings. We'll just go with how it works. Let's see. This is a TAC1 probe, right? It's a TAC1 probe where you order a company to synthesize the probe for you. I prefer to call this a probe because it is going to probe the reactions. Okay. What the company is going to do is going to decorate your sequence, whatever you are offering, with the two attachments. One is called R, another one is called Q. R stands for reporter, Q stands for quencher. Please follow this. The property of these two chemistry, these two chemicals, as long as this R is in a close proximity with the quencher, it cannot emit the fluorescence. In order to emit the fluorescence, the quencher has to be removed from the sequence. Is it clear? So there's day 0, you have the probe in your hand, much like the primer what you ordered. The probe is having a nucleotide sequence. On one side, you labeled it with reporter. Another side, it is labeled with quencher. Maybe labeling is maybe a kind of a confusing word. Maybe attached with a reporter. The other side, it is attached with quencher. So it's a nucleotide sequence. On one side, a reporter is attached. On the other side, it is a quencher is attached. It's a chemical that's given to you. Now you add this as one of the ingredient in PCR, much like the primers. Now what next? You have all ingredients, right? Plus one extra. What is extra? What is extra? The nucleotide sequence with reporter and probe attached. Let's see how it works. Very interesting, huh? Single-stranded DNA is formed after denaturation. Now what happens? This nucleotide sequence is designed from the complementary region. Correct? Much like primer, they will go and search for the complementary regions and bind also. Is it clear? The same way as the primer is going to do. Okay, no difference. Now, I think the next slide would be better. Read it. When intact, the fluorescence of the reporter is quenched due to its proximity to the quencher. That means there is no ground level fluorescence coming out of this setup. Now, probe hybridizes to the target. I already told you because of the complementarity. Now, you have the polymerase enzyme that's going to do the main job of adding the nucleotides. And that same enzyme is having another activity called 5' to 3' exonuclease activity. Right? So we are exploiting that activity to make the reporter fluoresce. How? It's quite simple. This enzyme will remove the quencher and reporter from each other. Okay? So it is getting released from the probe. It is coming into the reaction mixture as an independent inductease. Okay? The quencher is removed from reporter. Reporter is separated from the quencher. So every cycle passes, the probe is giving away the reporter and quencher into the reaction mixture. The moment reporter is removed from quencher, it gives fluorescence. Now you can imagine, as the cycle increases, there would be a progressive accumulation of what? Reporter in the solution. And also quencher, no matter, because they are separated from each other. Is it clear? As the reporter increases, the fluorescence will increase. The signal is proportional to the amount of amplified product in the sample. Is it clear? Yeah. This is a mechanism. The reporter is also called fluorophore. It's a quencher. Now what happens? The denaturation step. This is a probe what you added in the reaction mixture. This is a double-stranded DNA. In denaturation, what happens? They are separated from each other. Due to the complementarity, it will go and bind somewhere in the region where it has the complementarity. Now, two single-stranded, okay? And one of them would take out, would capture or complement with the probe. Now, this is what happening. The primer binding on the complementary strand. Now the primer come into picture now, right? And the moment primer comes there, what happens the next step? DNA polymerase comes. That is going to add the nucleotides. And on its path, it is going to encounter the fluorescence. Is it clear? On its path, it is going to encounter the fluorescence, the fluorophore. Here it's a quencher. So what happens? 5' to 3' exonuclease activity would remove this, and that is released into the solution. Is it clear? Now what happens? There would be a progressive accumulation of this. Yeah, they are separated. Okay. Now there is an increased accumulation of this particular fluorophore in the reaction mixture, and that finally it gives you the fluorescence. Is it clear? And ground level, there is no fluorescence whatsoever, right? As the reaction step increases, there is a, as and when the double-stranded DNA is accumulated, the 5' to 3' exonuclease activity would separate out the fluorophore or reporter from the quencher, and there is an accumulation of reporter in the reaction mixture, and collectively they will give you the fluorescence, right? Any doubts here? I'm going to stop it in another 5 minutes. Then we'll come back to the same slide after some time when we start the practical. Yeah. Any doubts here? These two techniques are somewhat opposite to each other. In the first dye-based, the bound dye is monitored. In Tachman probe, the released dye is monitored. The fundamental difference between these two. Is it clear? Did you understand the basics? Sure. In the dye-based, the minor groove is the one which is going to attract the cyber green. As and when the minor grooves are available in the form of accumulation of amplified product, you are going to get a fluorescence. That is a dye-based. In probe-based, what's happening here? There is a progressive accumulation of fluorophore or reporter in the reaction mixture with the help of the same TAC enzyme, which is going to add nucleotides that is going to remove with the help of 5' to 3' exonuclease activity. That removes the reporter from the nucleotide sequence from the quench. In the cocktail, you have everything. Since the fluorophore is not under the connectivity or proximity to the quencher, it is going to give you the fluorescence. Okay. So I really wanted to tell these two fundamental differences of a qPCR. The rest of the presentation is more with the practical, what you are going to do now. So do that, and we'll come back to the same slide, and from here on, we'll continue the presentation. Okay, I'll stop it here. And Shanu, what is the plan now? Are we going to do now or? Okay, discuss and let me know how much time it's going to take if you set up the PCR, 1 hour. Okay, please do that. Otherwise, we are not going to do it because we don't have time in the next 3 days. Okay. The practical what we plan today is expression of defense genes in plant. This expression is under the influence of a microbiome. Okay. The moment you release a microbiome on the plant, and the plant respond to that, and its a transcriptional profile is changed, and you are going to monitor with the help of some marker genes. I think the marker genes, what we have decided today is CERK, chitin elicitor receptor kinase. This is one of the marker for basal immunity in plants, and typically under the influence of the so-called microbe-associated molecular patterns. And we will do that practical. I think each group will take 2 time. There are 10 groups, and 2 groups can take 1 time point. I think we have a sample from 0 hour, 1 day, 2, 3, 4, and maybe up to 5 days are there. And each group can, 2 groups can take 1 time point. Okay. So the cDNAs are already, there are 2 other steps that we'll discuss in the theory portion. But the practical is all about setting up the qPCR leading up to the data analysis. Okay. So 2 groups, group 1 and 2 can take the 0 hour sample. 3 and 4 can take the 24-hour sample. 5 and 6 can take maybe 48-hour sample, and so on and so forth. Okay. And you do this qPCR. The machines what we have in the lab is a CFX96 by Bio-Rad. All reagents, ingredients, everything is arranged in the lab. And Shanu will explain here, then you set up. Maybe we'll have a break for lunch at 1:30. Okay. Yeah, please. Yeah, start your practicals. You are going to explain something here? Okay, fine. All right. You explain here and start the practicals. Okay. Yeah.
and we explain everything which we are going to do, the basics of what we are going to do. So in our practicals, what we have is our cDNA. That means from RNA, we have synthesized complementary DNA. From this cDNA, we are going to see the expression analysis. So what we have to do in this practical is, we have our cDNA, we have to dilute it, which is already done for you. Okay. Am I audible? Yeah, okay. So we have our PCR plates. So for this particular system of what we have in this practical lab is Biorad. There are many other systems like Roche and some other systems also there, but in this division we have Biorad. For that, there is a specific type of PCR plates which we use for this analysis. So what we are going to do, we have those plates, we have our template cDNA, and then this is the reaction primer you are going to use for the analysis. You can copy this. It contains nuclease-free water, master mix, which is a qPCR master mix. In this practical, we are going to use Promega. And we have forward and reverse reprimer. Those will be on the basis of the genes. One housekeeping gene will be using, that is actin, and the other reporter genes which we'll be using is PAD4 and CERK. So how you're going to perform? In single well, the total reaction mixture we will be having is 10 microliter. So every group has one column. So there are total 12 columns in the plate, and every group has to place their samples in these columns. 1, 2, 3, 4, sequentially you are going to do it. And the first 3 is the technical replicate for your first control sample. I will tell you which of those 3 which I'm going to provide you are the control ones, and the other one which is the treated one. Okay? The first one will be the control one, the next 2 will be your treated, first one will be control one, second to third will be your treated one. Okay? So first 3, you're going to use actin, next will be your defense gene, and the next 2 will be your negative reaction. Negative reaction means everything will be there except for the cDNA. So this is how you're going to proceed. We will seal those plates, and then we will keep those plates in the Biorad machine, and then we will proceed with the reaction. Any questions? I guess it's clear. Shall we proceed with the practical? Yeah, thank you.